Assalomu alaykum hurmatli tinglovchi. Men Buxoro davlat tibbiyot universiteti mikrobiologiya, virusologiya va immunologiya kafedrasi dotsenti PhD Mustafayev Feruza bo'lmoqdaman. Bugun biz ingliz tilida sizga leksiya o'tib bermoqchiman. Ya'ni ingliz tilidagi leksiyamiz bakteriyalarning, ya'ni fiziologiyasi haqida bo'lib o'tadi. Today we can study about the physiology of the bacteria and the growth, nutrient and metabolism of these all of the bacteria of the types. If you look at about the bacterial physiology, we can see neutron of these bacteria and environmental factors affecting survival of the bacteria, growth, multiplication, and metabolism all of these bacteria. If we look at about the neutrons, is neutron process. It's a process by which chemical substances called neutrons are obtained from surroundings environment, used for metabolic activity and growth for of the cells. And most of the medical impurity bacteria grows on simple medias with own source of organic materials too. Some bacteria require more complex media containing certain organic components and fastidose bacteria. Neutrons. If you look at some neutrons, bacteria is divided two big subgroups, for example, autotrophs and heterotrophs. And autotrophs is divided two subgroups. It's a hemoautotrophs and photoautotrophs. And heterotrophs is divided parasites, saprophyticus and symbioticus. Autotrophs, they are able to synthesize their own organic food from microorganic substances and can use carbon dioxide and carbon dioxide sides. Autotrophs is divided two subgroups, photosynthesis and hemosynthesis. Photosynthesis is a photoautotrophs and use energy from sunlight to produce organic molecules from the carbon dioxide and nitrogen dioxide for all of the growing plants depend on. Hemoautotrophs use an energy from oxidation of inorganic substances to synthesize organic components, and sulfur bacteria oxidizes sulfur to sulfur, and nitrosomes oxidize ammonium to nitrate. Heteroautotrophs and absorptive is divided saprophyticus, parasites, and symbioticus. And saprophyticus is a fetus on dead and declined organic matter, and parasites obtain nutrients from the host, and symbiosis, mutualism, beneficial associations. Optimal nutrient requirements for growth of the bacteria, which these need for bacteria. For example, first is water, source of carbon, source of nitrogen, inorganic salts, growth factors, and source of energy. If we look at, for example, first I told about the water. Water is the most important equipment, or as it's considered about the 18% of total weight. And this help elimination of all waste products, participation in metabolic reactions, and is integral part of the protoplasm. Source of carbon and energy. Bacteria are divided in two groups, and autotrophs and lithotrophs can use carbon dioxide as a sole source of the carbon for synthesis of all their organic metabolisms. And they require water, inorganic salts, carbon dioxide for growth, derive energy is from light, chemical, and other things. Heterotrophs or organotrophs bacteria unable to use carbon dioxide as a sole source of carbon, required in various organic from glucose, amino acid, nucleotides, and lipids. Source of the nitrogen is the atom of important biomolecules. For this is, for example, to growth like that. Inorganic salts, these are required for osmotic regulations. They are essential for certain enzyme system. The anion phosphate sulfate, the cation solutum, potassium, magnesium, iron, calcium, magnesium. Growth. Growth an increase in all of the cell components which ends in multiplication of the cell leading to an increase in population. It involves an increases in the size of the cell and increase the number of the individual cells. Bacteria is divided by binary fission. Growth factors. Growth factors is a need. Many pathogenic species require certain key substances for the growth factors or bacterial vitamins, and include B-complex vitamins, purines, pyramids, amino acids. They are provided by various body fluid, tissues, vivo, in the form of the yeast extract, blood and blood products in vitro, and other growth factors, for example, vitamin B complex, thiamine, riboflavin, and folic acid, and others. And we can see in here binary fission of the bacteria, how to divide it. For example, dividing methods of this. In picture we can see growth factors. Many pathogen species require the skin certain substances. And environmental factors. Environmental factors affecting growth of bacteria. And for example, what's the environmental factors? Carbon dioxide, temperature, pH, light, osmotic effects, mechanical and sonic stresses to affect. And in here we can see, for example, moisture and desiccation about the depend of the growth bacteria and carbon dioxides. And other requirements, we know about all of the bacteria divided about the 4 classes for example, obligate aerobics, obligate anaerobics, facultative anaerobics, and microaerophils. What's the mean? About look at is the obligate aerobics, it can grow only dioxide, and obligate anaerobics, they cannot grow about the dioxide, but they can grow only carbon dioxide. Facultative anaerobics, we can see they grow both of them. Microaerophils, it's grow only little % of the dioxide, about the dependent 18 or 11%. In here we can see pictures, obligate anaerobics, facultative anaerobics, obligate anaerobics, aerotolerant aerobics, and microaerophils, how to grow. About in picture we can see types of the growing about the depend of bacteria. Classification based on the temperature, we divided today to 3 subgroups. First, mesophils, and cyphrophils, and thermophilics. Mesophils grows about between 25 and 30 degrees. Is most of the pathogen bacteria depend on the mesophil bacteria. Cyphrophils, it's cold glowing, grows best below about 20 degrees, and thermophils is a depend of the high degrees. pH is a second another, for example, important things for growth to bacteria, and this is fall below about pH 6 and above 7.8. Its lighting is another some most important things for bacteria. Bacteria grows well in a dark, and some bacteria grow about some lighting environment. Osmotic effects, and we can today speak an important osmotic effects and mechanical and sonic stresses too for bacteria. Generation time. Some bacteria grows quickly. For example, we can see intestinal digestive system bacteria grow, for example, speedily, but some bacteria not grow speedily. For example, we can take about tuberculosis bacillus about take 20 hours to divide one bacteria to 2. Bacterial contents, the total content, viable contents, and the affected all of these. We can see this is all of these affecting these bacterial contents. Bacterial growth when the bacterium is added to suitable liquid medium incubated in growth follows and defined courses. Large phases. And we know about if we look at some about when divided bacteria to take some phases. For example, first lag phases, log phases, And in here we can see lag phases when bacteria is, for example, to cultivate some culture. We first can some lag phases, and after that we see these bacteria can divide so many numbers and take log phases, and in the stationary phases they can keep this number of these bacteria, and after that we can see phase of the decline. Today we finished our little of the lecture, and we continue our lecture to the next lessons about the physiology of the bacteria. Thank you for attention.